Hello everyone, I am Dr. Shraddha Sahu, Assistant Professor Biotechnology in Faculty of Engineering and Technology, Rama University, Kanpur. Today we are going to discuss on the topic enzymes. So what are enzymes? Enzymes, we can't imagine our life without enzymes. In biological world, enzymes plays a very important role. As we have heard about the chemical reactions, the chemical reactions may happen or may not happen in the absence or presence of catalyst, and so is the role enzyme plays in our daily life, because any of the biochemical reactions which are going on within the cells, these are not possible without the enzymes. So do you ever think what are the possibilities or what will happen if you don't have enzymes in your life? So enzymes, even the food we eat is getting digested with the help of a number of enzymes. So let us today discuss what are enzymes and how they function. So first of all, discussing about what are enzymes. So if we try to understand in a biochemistry, enzymes are the biocatalyst, because they are catalyzing the living reactions which are going on in the living systems, and also they are purified and obtained from living sources. They are not chemical molecules, rather they are biochemical molecules. What are biochemical molecules? These are the enzymes are the biomacromolecules. That means they are present in large quantities in living systems, whether it is bacteria, virus, plants or animals. So we can say, and all enzymes are proteins. All proteins are not enzymes, because proteins play structural as well as functional role, but enzymes are functional proteins. We can say they are specialized proteins. So therefore we call that enzymes are the proteinaceous biocatalyst, except ribozymes, because ribozymes are not proteins. These are the enzymes which are having other than protein molecules as a catalyst. So these are very highly specific. Now what is the function of an enzyme? So the enzyme catalyzes a biochemical reaction. It means it has some reactants. So here we call reactants as substrates. So these substrates are getting converted into product, and every substrate has its own specific enzyme, and every enzyme has its own specific substrate. It means the enzymatic reactions are highly specific, and the enzymes are such proteins which are highly specialized proteins. Next, we can say the enzymes have enormous catalytic power. So what is the catalytic power? How the enzyme is, because enzymes helps in biocatalysis. So it increases the rate of reaction. Enzymes, what is the rate of reaction? By lowering the activation energy. So what is the activation energy and what is the rate of the reaction? We will see it one by one. So here we can see rate means speed of the reaction. So we can measure the speed of the reaction by observing that how many molecules of substrate are getting converted into products with the help of a particular enzyme. So first of all, let us discuss the one more property of enzyme is they are not used up in the biochemical reaction, because what is the role of a catalyst? It is increasing the rate of a chemical reaction without being used up itself. So is the role enzyme is playing here. So it is not used up. After converting the substrate into product, the enzymes gets free for another reaction, and it goes and binds to the another substrate. So let us discuss the structure of a substrate. So coming to the substrate, Suppose this is an enzyme. Enzymes, how the enzymes convert the substrate into product? There is a particular kind of pocket within the enzyme. So this pocket is called the active site. This pocket is meant for the binding of the specific kind of substrates. As I told you, every enzyme has its own specific substrate, and every substrate has its own enzyme. So this specific substrate will come and bind into this pocket called the active site, and there is also in some enzymes there is another pocket or another site which is called the allosteric site. This allosteric site is not present in all enzymes but present in some enzymes. This is the site other than where the substrate binds, the allosteric substrates binds, other than the active site. So this active site is plays very important role in the conversion of substrate, in the binding of substrate, and how it works. Let us see at a glance. So this is active site at a glance. So what we can see is there is several kind of how the substrate and enzyme interacts. It interacts in a specific way. So what are those specific way? The substrate are binding with many weak interactions through the enzyme. So these weak interactions, so that the substrate and enzyme complex, when it forms, it gets stabilizes for a while, and that during the conversion of substrate to products, many chemical bonds are made and broken up. So this is a biochemical process. Suppose this is a substrate molecule. So this enzyme is having many kind of sites. This is a zinc binding site, this is the hydrogen binding site, as you can see this is the hydrophobic binding site. So there are different atoms and molecules, and this is the ionic binding site. Different atoms and molecules, they are having different kind of interactions with this enzyme, and this whole process happens or occurs within the active site of the enzyme. So this is how an active site of an enzyme plays a very important role. Now we can see what are the apoenzymes. So apoenzyme is the inactive form of enzyme, where the protein portion of the enzyme, where we have only protein portion of enzyme, and this enzyme does not act, or it cannot bind to substrate till the time some cofactor comes and binds to this apoenzyme. So what are cofactors? These are some inorganic elements. It may be some inorganic elements, and these are the non-protein portion. So it acts as an activator. So this cofactor comes and binds with this substrate, with this enzyme, and then the holoenzyme is formed. This is the whole enzyme, or we can say the active form of the enzyme. So this enzyme is now ready to attract the substrate. So the substrate will come and bind to the holoenzyme. So it will not bind to the apoenzyme. So this is the difference between the apoenzyme and the holoenzyme. So there comes another term known as the prosthetic group and the coenzymes. So what are prosthetic groups? These are nothing while the cofactor only. So if the cofactors are organic, like if they are organic and they may be permanently attached or temporarily attached, like they may be attached from covalent bonds or non-covalent bonds. So if they are organic, they are called prosthetic groups. Okay. So one very good example is the flavin adenine dinucleotide. So this prosthetic groups are the permanently attached. It means it is a part of the enzyme. The enzyme molecule is incomplete without this. So if the FAD cofactor is not present, some enzymes they have cofactors. So if the FAD cofactor is not present with that enzyme, this enzyme will not be functional without that. So so that's why prosthetic groups are also a very important parts of the enzyme. And then the coenzyme. So coenzyme part is the detachable part. A very good example is the NAD, nicotinamide dinucleotide. So These are also attached along with the active site of the enzyme. So, the substrate will only be able to bind with this enzyme molecule and getting converted into the product only with the presence of coenzyme. This happens with some of the enzymes. For some enzymes, the presence of coenzymes and prosthetic groups are very essential and important, as they are the integral part of that enzyme. This coenzyme may be detached. How? Because enzymes are the protein molecules and proteins are temperature sensitive. So, if we increase the temperature to very high, this may get detached, but the prosthetic group, in case of prosthetic group, it cannot be detached. So, now we will see the mechanism of enzyme action, how the enzyme acts. So, here we have two models for this. First was called the lock and key model. First model is known as the lock and key model. Here the enzyme acts as a lock and the substrate acts as a key. So, as we know that there is a specific key to every lock. Each lock is having its specific key. So, it means you cannot open the lock with a different key which does not belongs to a particular lock. So, is the case with the enzyme, because the enzymes are very specific molecules, substrate specific. So, this property of enzyme is called the substrate specificity. And suppose there are different substrates which are not structurally resembling the structure of the active site. So, apart from all those, a mixture of all those substrates, this enzyme will favor only this substrate, because this structure is resembling the active site structure of the active site. So, the interaction between substrate and enzyme will occur, and the enzyme substrate complex will form, and with the stabilization of this complex, and again the substrate will be getting converted into product. It means this reaction is going on. This reaction is made possible only when there is structural similarity between the active site of the enzyme and the particular substrate. It is not able, the enzyme is not able to bind with another substrate which does not resembles with the active site of this enzyme. So, this is and at last, when the product is formed, the enzyme sets free. So, this is the lock and key model. Another model, in this model it was assumed that the active site of the enzyme is rigid. It means it does not support the reaction if the substrate is not having if the substrate is little bit different. So, the reaction will not occur. It was supposed. So, this model was not practically accepted. Further, later on another model was proposed, which was called the induced fit model. According to this, what was the difference? Suppose this is the active site of the enzyme and this is the substrate. So, the substrate will come. When the substrate will come, there will be an interaction between the substrate and the enzyme. So, what will happen? This enzyme is not a rigid molecule. So, the active site will not be a rigid thing, because the proteins are of globular nature, and these are having three-dimensional conformation. So, there will be some change in the shape of the active site when the substrate comes and fits into this active site. So, then only there will be interaction between the substrate and the enzyme, and then it will be converted. Sometime this will be converted into enzyme product complex, and then the products will be released and the enzyme will be again setting free. So, the induced fit model was the most acceptable model, because it tells us that the enzymes are not rigid molecules, rather they are flexible, and with the interaction of substrate, when the substrate binds, it can be acceptable, and they can change their shape and conformation, and helps in the biochemical reaction to occur. Now, as I told you that the activation energy the enzymes lowers the activation energy. So, in the beginning of the presentation, I asked what will happen if there will be no enzymes, if there were no enzymes. So, this is the condition if there were no enzymes. This is the uncatalyzed reaction. Suppose we are taking some reactants. In this case, we are saying the reactants are our substrates. So, these substrates have some free energy, and this free energy, suppose we are taking here the reaction, suppose here we are taking the reaction progress, and here we are taking the Gibbs free energy on the y axis. So, this is the delta G represents the Gibbs free energy of the reactants when there is no enzyme, and this is the activation energy when there was uncatalyzed reaction. When you eat food and suppose there were no enzymes. So, in the absence of enzymes, it would happen that you are not able to digest your food through years long. So, without the presence of enzyme, this was the activation energy, and what does the enzyme do? This lowers the activation energy. This is the activation energy of the catalyzed reaction. When enzyme comes, it lowers the activation energy and the binding. So, who is lowering the activation energy? From where does this lowering of activation energy is occurring? So, here the binding energy is responsible. So, what is binding energy? The binding energy is the energy released due to the enzyme substrate interaction. When the enzyme and the substrate binds, there is an interaction. There is a they come to the close vicinity, and there is an interaction between the enzyme and substrate, and due to the binding, when this complex is formed, due to the formation of this complex, this binding energy is released. So, this binding energy facilitates the formation of this transition state, because this is not the final state. This is only the transition state. Why we are calling it as a transition state? Because after this, the enzyme will convert the substrate into product, and the enzyme product complex will formed, and the enzyme will be released, and the product will be released, and the enzyme sets free. So, this is the initial stage, this is the final stage, and intermediate state, this is the transition state. So, this binding energy facilitates the formation of ES complex and the transition state, and it also stabilizes the ES complex. So, this binding energy is the energy which lowers the activation energy. So, this is how there falls a difference between the catalyzed reaction and the uncatalyzed reaction. Suppose here are the substrates, and this is the free energy of the substrate. So, this will be the activation energy of catalyzed reaction, and this will be the activation energy of uncatalyzed reactions. So, we can conclude here that enzymes accelerate the rate of a biochemical reaction several times as compared to the uncatalyzed reactions. Okay. And next, the enzyme increases the rate of biochemical reactions by lowering these activation energy. So, the rate of biochemical reactions are enhanced number of times or many times, we can say, because without the presence of, what is the difference between the chemical and biochemical reactions due to the presence of enzymes in biochemical reaction is that in chemical reactions there are some spontaneous reactions which may happen due to without the absence or we can say without the presence of catalyst, but in biological systems, in biochemical reactions, the enzymes, without the enzymes there will be no reaction. So, they play a vital role, a very vital role and very important role in increasing the rate of biochemical reactions, and the enzymes does not change the position of equilibrium in a biochemical reaction. Okay. So, there are some factors which affect the enzyme activity. They are the temperature, pH, cofactors and inhibitors. Suppose some person is having fever. During that, during fever, the metabolism or the metabolic activities are reduced. Why? Because the enzymes are not able to work in higher temperature. during fever. So, this is a good example, and also in our body we have some sort of pH balance between the 6.9 or 8 to 7, 7.2. Okay. So, this pH is very important. There is always an optimum value for this temperature and pH of the for functioning of an enzyme. This is very specific and very different from for every enzyme. And now comes the cofactors and coenzymes. They act as an activators. Some enzymes are not at all able to work without the presence of activators and inhibitors. Also there are inhibitors. These are the molecules which may inhibit or which inhibits in reversible or irreversible manner the reaction, the enzymatic reactions. So, that is all for today's lecture. Thank you.